continue with the topics. I hope you are ready with another mechanism. I will not say hard, it's, and not really complete. It is a complicated, but I would say, let's say it is a fascinating process. Okay, so another examples of the fundamental knowledge that led us to change the world. Okay, before you're going to be the profound engineers, the technologists, okay, or great teacher. Okay, so this is the fundamental that you really know, that you really have to know. And this is the topics that the Faculty of Medicine really require us to always remind PPST that, okay, make sure the cellular respiration, the glycolysis process, the Krebs cycle, the future doctors to be really grab the points. Okay. And when I learned these topics when I was at UH, I remember that my lecturer will undergo one by one of the names of the process as we did for the photosynthesis. So this is I'm going to do with you, but it also has involved a lot of enzyme. So nama enzyme we don't have to memorize. Bagi yang masuk medik nanti hafal lah. Not in the asasi because I think it's unfair for those who are not going to medik. It's just only 50 of you. The rest of 200 of us will not go there. So you don't have to memorize the enzyme name, but the changes of each substance we have to understand. And even we don't have to memorize the name of the enzyme, but we have to understand the process, because this process, if you can see, it always compared with photosynthesis. So if we compare with photosynthesis, cellular respiration will be always applied according to, based on our health, because this process always happen in every cells, even in plant or even in animals. Especially in human, if there are slightly changes or interruption of this process, it may affect our health, or somehow explaining why this certain condition that we never experienced happen. Okay, so this process is actually really not really minor, but all the things that involve, especially all the protein, the enzyme, which is the protein which is very sensitive to temperature, high temperature and pH. So that's why this process is quite crucial. And I believe that in your semester one, you had learned about the PBL things, that when something's happen in the mitochondria might affect the development of the fetus, or you can have this child with underdeveloped. So it is very important. It's not only for me, for you. It's either you're taking or not the medical schools, but the knowledge of understanding respiration somehow may bring us to understand to what happened if there are changes in our body. Okay, so it will involve a lot of protein. So we understand that protein is very sensitive to temperature and changes, and we understand that protein enzyme has this mechanism inhibitions. So if things inhibit or bind to this kind of enzyme, it may stop the process. And understand that cellular respiration is far away from tarik nafas, hembus nafas. Okay, jangan imagine when we try to understand respiration, especially cellular respiration, it involves tarik nafas, hembus nafas. Okay, it involve tarik nafas, hembus nafas. Okay, cuba tarik nafas dulu dalam-dalam kelas. Okay, so you have supplied your body or your cell with oxygen. So that is the importance of that part of respiration. But we're not going to understand what happened if you tarik nafas, hembus nafas with your thorax of your lungs, things like that. But we have to understand that when we inhale, the oxygen that we had in our body will be used by the cell to produce ATP. Okay, so these are the importance of the process. Okay, so cellular respiration, the process that supply us energy. So the aim and learning outcome, the aim would be you must understand the aerobic and anaerobic respiration. So I would not try to relate this process according to any application, okay? But I would like you to really understand the mechanism. This is what really happened in our body, the chemical mechanism, so biochemistry. So this is things that happen in our cell. So you have to understand, able to explain and describe mechanism involved in both aerobic and anaerobic respiration. Able to write the net chemical equations, but I will give some discounts for this part, not necessarily because you learn organic chemistry this semester, so focus more on that part. Calculate the ATP molecule produced along the reaction. So this is very important. You must know how we can able to get 38 ATP and somehow we can get 36 ATP. So the ultimate goal for cellular respiration is produce ATP. I had give you a brief explanation about ATP previously. It was adenosine triphosphate. Adenosine yang ada 3 phosphate, so it may somehow turn into ADP or AMP, adenosine diphosphate and monophosphate. Is it us human really need for energy? So where we can get the energy? So this is the fruit that supply energy that we get from photosynthesis. So the energy required for metabolism, movement, active transport, cell maintenance, production of important substance for another mechanism, transmission of impulse. So this is another important topics that you will learn after my topics. Without some energy, you may not able to respond. Ini saya pernah explainkan hari itu kenapa kalau you drink alcohol, you may get hungover and your movement might be abnormal, not as fast as like the normal. So that is related with this process, and the energy is for control the body temperature. And we try to relate the situation where glucose is produced from photosynthesis, while here in the cellular respiration, glucose is breakdown for respiration to get energy. Okay. And there are 2 forms of having this energy, is from aerobic and anaerobic. Okay. And if you can see, photosynthesis occur in chloroplast, while you had learned from even in your high school, the powerhouse, the source of power, the cell that provide energy is mitochondria. So this is the moment along this while you know that mitochondria is the power of the house of power that supply most energy. Now is the time that we have to really understand how the energy produced. Okay, Nur Faiza, I think you accidentally mute your microphone. Please unmute. So please mute your microphone, Nur Faiza. Thank you. Okay, for cellular respiration, okay, we will see a lot of oxidation and reduction process. So this is a really chemical process where we have this glucose will be oxidized and producing CO2, and the oxygen that we inhaled just now will be reduced and will bind with H+, that abundance in the cell to produce water. So instead of having photolysis or lysis of water, we will having a make-up process of producing water and energy. So you will have these words of oxidation, reduction, decarboxylation, removal of carbon dioxide, phosphorylation, adding phosphate. So we had learned this before in phosphorylation. So here in this cellular respiration, it's required no lights or do not have photoactivation, it will able to do phosphorylation, meaning add on phosphate. And isomer, where some molecules will change to another structure, but memang dia. So I would like to give you a brief or overview of the process from this video.
How do you turn that bite of food into a chemical that a cell can recognize and use as energy? The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistants. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins.
Is it lagging very bad, class?
No.
Okay, let me continue. Sekejap,
The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide. How do you turn?
Okay. So from the video,
How?
okay, from the video, okay, I need you to turn to page 205 for summary. Okay. So mari kita baca sama-sama. Okay, kalau tidak sempat, sekejap saja ya. Saya sudah copy ni. So saya mau kita semua baca the summary from the textbook that I had given in the Telegram. Okay. So let us give 3 minutes to finish with the reading. Okay. So from your reading, we understand that I would like you to see the comparison between photosynthesis and the respirations, where the glucose that we add is produced in the photosynthesis from the light-dependent reaction, where it produce ATP and NADPH that will be further used in dark reaction and producing glucose, and the glucose will be used up in cellular respiration, especially during the glycolysis process, and it ends up producing 2-pyruvate, ATP and NADH. Okay, there is a slightly difference between NADH and NADPH. So only available in plant, and NADH is only available other than plant. But plants also do have NADH. And then these items will be further used up in Krebs cycle, and then will be producing powerful energy electron carrier, which is NADH and FADH2, that we don't found this in plant in photosynthesis, and this will be function in the electron transport chain and producing ATP through chemiosmosis. So I really hope, I strongly believe you really understand well the chemiosmosis process, where this actually, the similar process happens in cellular respiration. But the names of the electron transport chain is a bit longer, more than what we had in photosynthesis. So the difference between the two processes is This is a making up process. This is catabolic or breaking down. It occurs in plants, in both plant and animals. Require lights, not require lights. Happen in chloroplast is mitochondria. Glucose is end product. Glucose is starting product. CO2 and H2O is starting product, and oxygen is given out. Oxygen is taken in, and CO2 and H2O are end product. Energy is trapped. Energy is released. Occur mainly during the day. Continuous day and night. Selagi kamu tak berhenti makan, selagi itu dia ada respiration. Light energy converted into potential energy, while respiration converted potential energy into kinetic energy. Take place in presence of catalyst. No catalyst is needed for respiration. The day weight of the plant increased due to synthesis of food material. Plant decreased due to utilization of food material. Okay. Now let me introduce to you to the energy carrier that will helps most of the process of making ATP. So the first one is NADH. This is the difference between the NADH and AD plus, where it has one, while the other one is reduced. Okay. If you can see that the full name for NAD plus, okay, nicotinamide. Nicotinamide, okay, nicotinamide, adenine dinucleotide. Adenine. Kalau tengok, okay, which part is adenine dinucleotide? What is the functional unit for nucleotide? It must have nucleus, it must have pentose sugar, nitrogenous base, and phosphate group. So it has di. So it should have two of these things. So the first one and the other one connect by condensation. Ya, ikan macam glycosidic bond tu condensation. So it has these two. So this part is adenine. Is this adenine similar to the adenine that we learned in nucleic acid? Yes, it is the same adenine. So adenine, okay, adenine, is it purine or pyrimidine? Okay, saya pun nak ingat. So macam tu lah. So it has connection. So this is pentose sugar. NAD plus act as a hydrogen acceptor. So dia akan bind di sini ya, yang dia akan bind dengan oxygen. Derived from nicotinic acid of niacin or vitamin B3. Source meat, wholemeal bread, liver, yeast extract. So that is NADH. Another one is FADH2, reduced form of FAD plus. The full name is flavin adenine dinucleotide. So kita ada dua nucleotide juga. Sorry. Now, first nucleotide. So another nucleotide. Okay. Produced in Krebs cycle. FAD plus is ADP molecule bound to riboflavin or vitamin B2. So ini lebih kurang kita cakap ada coenzyme. So most of the enzyme, vitamin biasanya act as a coenzyme. So FAD plus act as a hydrogen acceptor. One molecule of FADH2 yields two molecules of ATP as it adds electron to the ETC at lower level of NADH. So the most powerful energy carrier is FADH or FAD plus. So sumbernya, the source of, you should always have a B3. So nampak tak relation situ tu? Sumber vitamin B complex. Yang nak makan sayur, tak dapatlah vitamin ni kan. Daging. So as simple as that. So that's why cellular respiration ni saya tak boleh explain dia punya advanced technology. But then if you are lacking some of it, there will be a problem to your how our body will respond. Okay. Another one is I forgot to explain about the ATP. So if ATP is actually adenosine, adenosine is also adenine that also have pentose sugar with 3-phosphate group. So let's have a look on the aerobic respiration. You read that it has glycolysis, and then we had Krebs cycle, and we have ETC, electron transport chain, or it's also called oxidative phosphorylation. Krebs cycle, in your books, it not say 3 stages, but some books may refer 4 stages. So it is actually because before it goes to Krebs cycle, the pyruvate, which is the main output of the glycolysis, have to be attached with coenzyme A so that it will able to enter the Krebs cycle. So the process is called link or chain reaction. Chain reaction, or some books call link reaction. So link reaction meaning that it have to modify the pyruvate. So kena faham glycolysis occur in cytoplasm. So this Krebs cycle occur in mitochondria. So the other part is inner matrix, dalam matrix mitochondria kan. So the changes of places somehow require changes of identity. Contohnya macam saya, nama saya di Jepun kena panggil Siti Sang. Contohnya, Siti Nurusha Pasang. Contohnya kan, orang nak panggil Siti Nurusha pun nak pandai. So, adakah Siti Nurusha Pasang and Siti Nursyafa di Malaysia dengan di Jepun tu sama? Yes, orang yang sama. Cuma different tempat nama dia lain. So, same goes to the pyruvate. Okay, pyruvate is cytoplasm. So in order for the process to be continue and move from other places to another, especially to the mitochondria, so it has to do under some adjustment, bind with the acetyl-CoA and then able to do the Krebs cycle. Okay, itu maksudnya. And we will start with the glycolysis, where it is a breakdown of glucose into 2 molecules of pyruvate. So the M is glycolysis is pyruvate. So is this the similar pyruvate that we found in the C4 plant? Yes, it is the same things. Some books call citric acid cycle, some call Krebs cycle, but biasakan panggil Krebs cycle. So Krebs cycle ni ada Uncle Krebs macam Uncle Kelvin juga yang discover the cycle. So oxidative phosphorylation or the ET, electron transport chain movement through osmosis. Okay, bersedia untuk pembelajaran yang lebih complicated. Jangan tertidur, bangun. Okay, you will learn glycolysis, link reaction, or another name is decarboxylation of pyruvate to acetyl-CoA, and then the transition reaction. Okay, then citric acid or Krebs cycle, oxidative phosphorylation. Okay. So link reaction maksudnya decarboxylation of pyruvate to acetyl. What does it mean by decarboxylase? Tolak CO2. Okay, so this is the overview of the location of the process. So we have cytosol or the cytoplasm. Okay, kalau kamu ingat bila kamu lukis sel, you have this, besar juga kita punya nukleus ni. You have this nucleus, you have this ER, you have this mitochondria. So yang di luar-luar ni adalah cytoplasm. So you have this microtubule, kan? Macam-macam lah. So this is the mitochondria. So the glycolysis happen here, cytoplasm. And the output of the pyruvate have to do some adjustment, which is link reaction to enter mitochondria. So glucose produce pyruvate and other stuff, and then pyruvate will be oxidized, decarboxylation and producing acetyl-CoA, and will be able to enter Krebs cycle and producing, here we will produce a lot of NADH as well as FADH that will be further used up in this area. And where will be the abundance of H+? The matrix. So due to the concentration gradient along the movement of the electron will cause the H+ to going out, balancing the electrochemical through ATP synthase. That will trigger ADP and phosphate that free around ATP synthase to bind. Okay. Faham ah? Okay. Masih lagi bersama-sama saya kelas?
Yes. Masih.
Okay. So for ATP generation throughout cellular respiration process, where we have this oxidative phosphorylation and substrate level phosphorylation. For oxidative phosphorylation, okay, this is through chemiosmosis process. Okay, so let's have a look for the first one. So the Krebs cycle and the ETC we will continue next week. So we will going on with the glycolysis in these few minutes. Okay, glycolysis from the word lysis, splitting of glucose. Not really split. Okay, break down glucose into two molecules of pyruvate. So glucose, we understand it has 6 carbon, so pyruvate must be 3 carbon molecule. Okay, sama. Macam saya explain tadi, we have 6 carbon split into 2 menjadi pyruvate. Location, cytoplasm, stages, energy investment phase and payoff phase. Net reaction, glucose require NAD plus phosphate ADP and producing pyruvate NADH that will be used in Krebs cycle. Will produce 2 ATP net H plus and H2O. Glycolysis occur whether or not oxygen is present. Okay, macam mana kita mau dapatkan? So kita akan tengok macam mana kita dapatkan pyruvate. The 3 carbon molecule. Okay, and ADH will be converted, not will be converted, actually will be used to produce ATP. Okay, so according to your textbook, page 190. So these are the 10 steps where from glucose, it's finally producing pyruvate. So 10 step ni kena ingat lah. Ini sama kamu ingat. Tapi nama enzim ni tidak perlu tidak apa. Biar budak medik siapa nanti masa dorang sama medik. Okay. Okay, glucose. It will go phosphorylation. Anything processed that add on phosphate is a phosphorylation. Huh? Glucose have to add phosphate here. So this is our glucose. Have to add phosphate. So adding of phosphate perlukan ATP. So glucose becoming glucose 6-phosphate. Dengan saya dulu, kita ingat nama dia. Glucose turn into glucose 6-phosphate and then it became fructose 6-phosphate. Okay, so dia pakai isomer. Fructose 6-phosphate and then it fructose 1,6-biphosphate. Okay, sudah ingat? And then menjadi glyceraldehyde triphosphate ataupun G3P. Okay, daripada glucose menjadi, ini saya sedang menghafal juga ni ya, glucose 6-phosphate and then menjadi fructose 6-phosphate and then it becomes fructose 1,6-biphosphate. Okay, and then it become glyceraldehyde 3-phosphate. Itu je, senang kan? Apa saya? Okay, sebenarnya fructose 6-phosphate ni masalah lah kan, bio ni unstable je. Okay, fructose 1,6-biphosphate akan bertukar, will be changed into dihydroxyacetone and glyceraldehyde 3-phosphate. Unfortunately, dihydroxyacetone, okay, dihydroxyacetone will always unstable ataupun we would say unstable lah. So instead of macam ni saya cakap fructose 1,6-biphosphate will always turn into 2, dihydroxyacetone phosphate and G3P. But dihydroxyacetone is always unstable and will tend to change into G3P. So here we're going to have 2 molecule glyceraldehyde. So instead of from here 1 and then now it become 2, it should be 1 and 1. But this one always tend to change into this one. So in the process, we will any books will always refer to have 2 glyceraldehyde 3-phosphate. Okay. But this substrate, dihydroacetone, will always be produced by fructose 1,6-biphosphate. Somehow, for the unexplained condition, contohnya contohlah ada benda yang bind dengan dihydroacetone ni, contohnya mungkin cyanide kah, mercury kah, dia bind dengan dihydroacetone ni menyebabkan tiba-tiba badan kita lumpuh ke apa. That kind of situation that may explain reaction yang mungkin disebabkan oleh reaction of this item dengan benda-benda lain. Okay. So walaupun dia ni sepatutnya dia ni tidak stabil, always tend to change into glyceraldehyde triphosphate, but it always be produced. Okay. In normal situation, it will be isomerase by using this enzyme. Always using isomerase to be in a glyceraldehyde triphosphate. So from here, so this okay, let's have a look what are the stages. Okay. So from here, the G3P will become 1,3-biphosphoglycerate. So similar that we found in the phosphorus-Calvin cycle. 1,3-biphosphoglycerate and become 3-phosphoglycerate and then 2-phosphoglycerate. It changed into phosphoenol pyruvate and pyruvate. Boleh? So from G3P, it became 1,3, ini satu phosphate, 1,3-biphosphoglycerate, and then 3-phosphoglycerate, and then 2-phosphoglycerate, phosphoenol pyruvate, and then menjadi pyruvate. Hafal? Hafal sudah ke? So, okay. Here, the first one, from glucose to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-biphosphate, and then it becomes dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So this process we call as energy investment process. Kenapa energy investment? Sebab glucose kalau mau jadikan dia phosphate, kita tambah phosphate. Jadi kita perlukan ATP. So itu dia panggil perlukan energy. So investment of energy. So dua tempat yang perlukan energy, from glucose to become glucose 6-phosphate perlukan ATP, and then from fructose 6-phosphate becoming 1,6-biphosphate perlukan ATP. And the other part is between 1,3-biphosphoglycerate to 3-phosphoglycerate perlukan, it's producing ATP. This here, it's called payoff because it's producing ATP. Okay. So we will have oxidation process, substrate level phosphorylation, isomerization, and dehydration. So kalaupun saya keluarkan dalam soalan lah kan, macam objektif lah. Saya tidaklah macam okay, apakah nama seterusnya. Biasanya kita akan macam bagi ada blank area, so you have to complete what would be the following substrate. Okay, so from the glycolysis process, we use up to ATP, but along the process, we able to produce 2 pyruvate, 2 ATP, 2 and 8. Actually, we able to produce 4 ATP, right? But consider that we had used 2 ATP, so tolak. This ATP will minus the first 2 ATP that had been used. And the total net of ATP from the glycolysis process is actually 2 and 2 NADH. So these are the output of glycolysis. Okay, kenapa ada dua pyruvate? Di sini ya. Started from here. Here. Okay. Fructose 1,6-biphosphate will produce, this is, we would say like 6 carbon compound and becoming 3 carbon compound. So this is the similar process. So we have 2 molecules. So from 1 molecule, it becomes 2 molecules, but less carbon. This is 6 carbon, this is 3 carbon. Okay. Okay, so finally, the output of glycolysis is pyruvate, ATP, and NADH. Jangan tersilap to NADPH ya. So ini hanya dalam plant. In cells we have NADH. In cellular respiration. Okay. Boleh sampai situ kelas? Faham? Kelas?
Boleh.
I need some feedback. Faham kah?
Boleh, Miss.
Boleh, tapi tepuk sudah kan? Okay, so I hope you find this topic interesting or fascinating. Okay, I would say fascinating process. Okay, if you can see that how I can develop the knowledge from biocatalyst to photosynthesis, but this one I couldn't really find the best way of explaining it other than you really must know the mechanism. Okay, sebab kesan dia adalah penyakit ataupun the movement that might changes to our body. Okay, but by understanding that, this also encourage us to produce or to establish development of new drugs. Okay, ataupun ubat-ubat herba ataupun drugs that maybe help us, especially in our health. Okay, so with that, I end our lab. I end our lecture session and hope to see you next week for the final session with me on this cellular respiration. So saya harap semua orang bersemangat minggu hadapan sebab